A very good morning to one and all. Respected dignitaries present for today's function, guest speaker Dr. Shagufta Parveen ma'am, founder chairman Dr. Sames Dr. Vinod Bhandari sir, Chancellor Sri Aurobindo University Dr. Manjushri Bhandari ma'am, Vice Chancellor Dr. Jyoti Bindal madam, all the HODs of various departments, faculty members, and dear students. Myself, Dr. Deepa Nair, Associate Professor, Department of Physiology, take this opportunity to welcome you all to one of its kind CME entitled Novel Insights and Uses of Regenerative Medicine, organized by the Department of Clinical Physiology at Sri Aurobindo Institute of Medical Sciences, Indore. I invite all the dignitaries present here to light the lamp and grace this occasion. Surgery. Now, I request Dr. Jayashree Tapadia Madam, Professor and HOD, Department of Physiology, Convener, Curricular Subcommittee Phase 1, Sri Aurobindo Medical College, who is the organizing chairperson of this CME, to introduce our guest speaker for today's event. Dr. Jayashree ma'am is a person whose strong will and determination has led to the development of state-of-art skill lab at our institute. She is an inspiration for all of us and has always motivated and encouraged us towards clinical physiology and has many research publications to her credit. She is an athlete herself and has always encouraged others to lead a healthy life full of zeal and enthusiasm. Over to Dr. Jayashree ma'am.
Thanks, Deepa. Good morning, everyone. Respected dignitaries on the dais, Founder Chairman, SAMES, Dr. Vinod Bhandari, sir. Chancellor, SAMES University, Dr. Manjushri Bhandari, ma'am. Vice Chancellor, Dr. Jyoti Mindal Madam, all the HODs and all my faculties. It's my pleasure to introduce the guest speaker for today's event, Dr. Shagufta Parveen. Dr. Shagufta Parveen is presently working as an assistant professor at Manipal Institute of Regenerative Medicine's MAHE. She has PhD in stem cells, biology and cellular reprogramming, MSc in microbiology, and has over 21 years of postgraduate teaching experience. Her specialization or research areas include stem cells biology, cellular reprogramming, induced pluripotency, developmental biology and genetics, modeling, early human development, and disease using the pluripotent stem cells, diseases modeling, and drug discovery. She is also a MHE recognized PhD supervisor. She has received research grants from several funding agencies such as DBT, Department of Biotechnology, ICMR, DRDO, etc. over 2 crores. Dr. Shagupta is also recipients of many awards and honored, and also has published in several high impact factors journals of national and international repute. On behalf of the Department of Clinical Physiology, SAMES Indore, I would like to welcome Dr. Shagupta for her CME talk on regenerative medicines. Thank you.
Thank you, ma'am. Now I request Chancellor Sri Aurobindo University, Dr. Manjushri Bhandari, Madam, to welcome our guest speaker with a floral bouquet. Thank you, madam. I request our guest speaker to take over the session. Ma'am, please.
Greetings to all of you. I am Shagufta. I come from an institute called Manipal Institute of Regenerative Medicine. It is in Bangalore. We belong to a university or deemed to be university called Manipal Academy of Higher Education. I will be presenting a talk called, I've named it as Stem Cells, Progress and Promise in Curing Diseases. I would like to start off by first defining a stem cell. A stem cell has it's a cell which has two major properties. The first one is that it can self-renew itself. That means it can divide to give a cell that is identical to itself, and it has immense differentiation potential. That means it can differentiate into cells of different types of different organs of the body. So any stem cell should have these two properties. It should be able to self-renew, and it should be able to differentiate into different types of cells. Without these two properties, it is not defined as if a cell has anything else, if it does not have these two properties, it cannot be defined as a stem cell. Now, depending on now these stem cells, they can be isolated from the embryo, and you can get them from adult tissues and organs. So there are two sources of these stem cells which are there. Now, depending on the potency of the stem cell, we classify them as totipotent cells. A totipotent cell is something that can give rise to any part of the body, all cells of the body. A pluripotent cell is which can divide again to give all embryonic lineages. A multipotent cell like the hematopoietic stem cell, which can give rise to certain lineages, and unipotent stem cells, which give rise to only one type of cell. I would also like to define two terms over here. One is reprogramming. Reprogramming is an experimental procedure by which we increase the potency of a cell. You may understand it a little later when I tell you. And then there is a transdifferentiation plasticity, where one cell can convert into another type of cell. There are lots of adult stem cells which are present in our body, and these adult stem cells, they basically are involved in repairing any damage that happens during the course of our lifetime to organs which are present in the body. Usually these adult stem cells, they're quiescent. That means they don't do much. They keep, what happens is if there is any damage, only then they get activated and they replace those damaged tissues. Now, this concept of stem cell, it basically began with the hematopoietic stem cell. All of you being in the medical field are very well aware of the hematopoietic stem cell. And it was the first type of stem cell that could be transplanted and it could be transfused. So the whole concept of stem cell therapy began with this hematopoietic stem cell. There are a few pioneering scientists which recognize this potential of the hematopoietic stem cell, and they started doing these transplantations and translations with the hematopoietic stem cell. The first one of them is Arthur Pappenheim. Important ones like Alexander and Ernst Newman. All of these scientists, they recognize the importance of, they recognize that the hematopoietic stem cell could divide to give rise to different blood lineages. Now, what happened in 1981 was that there were two scientists by name Martin Evans and Kaufman. What they did was they isolated pluripotent stem cells from the mouse blastocysts. So they isolated embryonic stem cells. After a few years, another scientist by name James Thomson, he isolated embryonic stem cells from the human blastocysts. Now, these stem cells could be differentiated into any type of cell of the body. So you could make neurons out of them, or cardiomyocytes, or liver cells, or anything out of them. Now, this diagram over here, it shows that these embryonic stem cells, if you keep them for long enough, or if you treat them with certain chemicals in the laboratory, they give rise to any type of cell of the body. Now, after isolating these cells, these scientists understood that these cells had a potential to regenerate different parts of the body. So they were understanding that it could make any of the body cell. Apart from them, there were other scientists who thought a step ahead, and they said that these cells could regenerate any part of the body. So if you have a degenerated brain, they said we can incorporate these cells into the brain and it is going to regenerate the brain. That is a little far-fetched imagination so far right now at this point in time. But yes, everybody is working towards regeneration of degenerated organs using stem cells. Now, when they isolated the embryonic stem cells, after a few years, there were some controversies which came into being, saying that embryos could not be used to generate embryonic stem cells. They considered that embryos are living, they considered embryos as potential human, and they said we could not use embryos to isolate embryonic stem cells. And there was a little bit of controversy in most of the countries. So to use embryos, people had to go through a lot of approvals and ethical clearances. By this time, what happened was there were two scientists by name Gordon and Yamanaka. They devised a method by which you could take an adult cell and convert it into a pluripotent stem cell, which was very similar to an embryonic stem cell. Now, this was a very, very important discovery, or a very important finding that these two scientists did. Has anybody heard of these induced pluripotent stem cells? The students here? No? Has anybody heard of any stem cell, embryonic stem cells? You are not interacting with me. Has anybody heard of embryonic stem cells? Yes? What do you think they do? Do you think they are in a in vitro or in vivo condition? Okay. Now, according to Yamanaka's procedure, if you can take any cell of the body, any fibroblast, blood cell, skin cell, or any other somatic cell, and these cells are introduced with 4 genes. These are pluripotency related genes. When you introduce or transfect these cells with these genes, these adult cells, they convert themselves or they reprogram themselves into embryonic stem cell like condition. These are called induced pluripotent stem cells because we are inducing them to become pluripotent. Now, after this, these cells can be differentiated into any cell type of the body. So now they behave very identical to embryonic stem cells. In culture, embryonic stem cells grow like these as colonies. We give them feeders upon which they grow as colonies. Induced pluripotent stem cells are very similar to these, and they also grow like colonies. Now, the advent of induced pluripotency was so basically, if you take any pluripotent stem cell, be it an embryonic stem cell or an induced pluripotent stem cell, it can differentiate into any cell type of the body. If you give them proper growth factors and cytokines in vitro, they become any cell type of the body. And the vision of scientists is that these cells can be transplanted into human beings, and they will be they can regenerate any degenerating organ in the body. Now, apart from pluripotent stem cells, there are adult stem cells which are also present in the body. Every organ has some kind of adult stem cell, like bone marrow has the hematopoietic stem cell, kidney has stem cells, pancreas, adipose tissue, skeletal muscles, brain, liver, heart, all of them have a stem cell. These stem cells remain quiet. When there is injury, they divide and they try to replace the dead cells with the good ones. Apart from these adult stem cells, there is another one very important stem cell called the mesenchymal stem cell. The mesenchymal stem cell is a kind of a stromal cell which is present in most of the organs. And these mesenchymal stem cells are also very important in regenerative medicine. Basically, the mesenchymal stem cells can differentiate into osteocytes, adipocytes, and chondrocytes. And if they are coaxed enough, they can differentiate into other types of cells. Now, so far, it's all very fantastic, and everybody believes that regenerative medicine could be a reality in the near future. We just have to differentiate cells into one type of body cell, and we can put it back into the patient, and it is going to just go and regenerate whatever is missing from the patient. But there are major hurdles when it comes to these stem cells. First of all, we will have to decide whether the stem cell that you're going to use for treatment is going to be autologous or allogenic. Now, if there is a genetic disease that you're trying to correct, it does not make any sense to use autologous cells because the autologous cells are going to have the same genetic problem. And so it is obvious that we want to use cells from another patient. But if you're going to use cells from another person, the problem will be immune response because the patient is going to mount an immune response against the foreign cells. So these are the two major, this is the decision that needs to be made, whether we are going to use autologous cells or we are going to use allogenic cells. If you're going to use allogenic cells, we have to find a method by which they do not mount an immune response. Today, we are making engineered cells called universal cells. These universal cells do not have any MHC complexes on their surfaces. Thereby, when transplanted, they are not going to mount an immune response, and they might be useful in stem cell therapy. Apart from this decision, there are three major problems which there are three major drawbacks of stem cells. The first one, of course, is immunogenicity if you're using from another person. The second problem is that these cells do not integrate into organs of the patient. Like, for example, if you inject a neuron into the brain, it is not going to go and integrate successfully into the circuitry of the brain neurons. It is going to not, if there is misintegration, it is going to fire abnormally. If it is going to fire abnormally, it is going to lead to a whole nother problem. Likewise, in cardiomyocytes also, they do not integrate into the circuitry of the cardiomyocytes. So that's one major problem. Though we can inject them and into the organ or into the body, the chances of these cells integrating into the tissue architecture is very less as of now. The third problem is tumorigenicity. Pluripotent stem cells are known to form tumors inside the body. So they cannot be used easily. And even mesenchymal stem cells sometimes cause tumors in the body. So if you're injecting or if you're going to use them for regenerative medicine, you will have to first make clinical protocols and experimental protocols which can handle these three problems. And now I spoke of three types of stem cells: the embryonic stem cells, adult stem cells, and induced pluripotent stem cells. I've just classified them. The source of tissue from which they are isolated. The embryonic stem cells, of course, come from the embryos. Adult stem cells come from postnatal sources, and so do induced pluripotent stem cells. According to the differentiation potential, embryonic and induced pluripotent stem cells are pluripotent, like their name says, but adult stem cells are multipotent. That means they can differentiate into a few cell lineages, but they cannot form all cell lineages. According the lifespan of pluripotent stem cells is much high compared to adult stem cells. The immunogenicity is dependent, like sometimes it is allogenic and sometimes you can use autologous cells. Tumor formation, the pluripotent stem cells do form tumors, whereas the adult stem cells don't form tumors mostly. And clinical application is with differentiated cells. You can directly transplant adult cells into the body. And again, they are with differentiated cells. Can anybody tell, when I'm saying differentiation, can you understand what I'm saying? The lady first. Yourself. Do you understand what I'm saying by the word differentiation? Can anybody? What do you understand when I'm saying differentiation? Nobody? What do you think differentiation means?
मैम जब सेल्स स्पेशलाइज होती है कि इस सेल को आगे चल के लंग्स ही बनाना है और इस सेल को आगे चल के लीवर ही बनाना है, तो दैट इज कॉल्ड डिफरेंशिएशन.
दैट इज कॉल्ड डिफरेंशिएशन. कैसे करते हैं डिफरेंशिएशन? मतलब लैब में होता है या बॉडी में हो सकता है?
बॉडी में होता है.
बॉडी में तो नेचुरली होता ही है, लैब में कैसे करेंगे?
आई डोंट नो, सॉरी.
वी कैन ऐड जो कोई फैक्टर बॉडी में उसको डिफरेंशिएट करता है, अगर हम लोग वही लैब में रेप्लिकेट करते हैं, मतलब लैब में डिश में अगर आप वही फैक्टर ऐड करते हैं, देन दे डिफरेंशिएट सिमिलरली टू व्हाट इज हैपनिंग टू द इन द बॉडी काउंटरपार्ट. आई होप come on, if you don't interact, बहुत boring हो जाएगा. So please interact with me. Okay, so now these stem cells, like I told you, when they were discovered and as time is passing by, they are becoming very, everybody is very curious to know what can we do with these stem cells. So first of all, they are a fantastic research tool. You can do research on disease, on development, on differentiation, And you can find out, like you can research everything that happens in the body by using these stem cells, because they are replicates of the bodily cells in the dish. We can do excellent research with these stem cells. You can screen for toxins. Now, normally when we are doing toxin screening, we use animal models. Animal models are fine, but they do not recapitulate human situations completely. A mouse is still a mouse and a human is still a human. So if you use human cells for toxicology studies, the results which you get are more relevant to the human situation. So use of pluripotent stem cell in toxicology studies is also very popular, and it has given results and insights which could not be achieved by doing animal studies. You can look for new drugs. Like for many diseases, scientists have used pluripotent stem cells, and they have identified or discovered drugs which are much more effective, which they could not identify by using animal models. You can use them as drug delivery systems. You can use them for tissue engineering. Like if you have a neuron or if you have a bone cell, and if you integrate it with the tissue engineering procedures, you can make a scaffold which has human cells into it, and it is going to integrate into bone or any other organ much more better. And the final goal is the ultimate goal of stem cells is to use them for cell therapies, where we can just use them to regenerate or cure diseases, regenerate degenerating organs and cure diseases. Now, these pluripotent stem cells, like I told you, in today's time, we use more of iPS cells, that is induced pluripotent stem cells. We do not use embryonic stem cells more because I told you that there are ethical concerns. We just cannot take embryos, isolate cells from them and use them for research. Because what happens is a lot of exploitation happens during the course, and overall to use an embryo which could become a potential human is not allowed. So the more widely used pluripotent stem cell type today are the iPS cells. Now, iPS cells, they have advantages over embryonic stem cells. They avoid the use of human embryos, which are required for embryonic stem cell derivation. They can be used in basic research. You can produce new disease models from them. For example, you can take a somatic cell from any genetically diseased person, Alzheimer's, Parkinson's, schizophrenia, cardiac diseases, thalassemias. Any of these patients can give their somatic cells. These somatic cells can be genetically manipulated and converted into pluripotent stem cells, and those pluripotent stem cells can be used to model the disease. This is a very, very effective way of modeling diseases today, and it is giving information which could not be achieved, which could not be gotten earlier previously with other model systems. They can be used in drug development and screening. They can be used to make disease-specific iPS cells. Now, with these disease-specific iPS cells, like you can model the disease, you can find new drugs which are effective against those diseases. They can be used to study human development and disease at a molecular level. And of course, finally, we use them for the ambitious goal of cell transplantation. And indeed, it is the iPS cells which have gone into the first human transplantation. It was used to treat macular degeneration in Riken Institute, Japan. Now, over the world, people have understood. Most of the stem cell scientists consider iPS cells as a very important resource, and there are lots of repositories across the world which are banking these cells. These cells are basically made from diseased and controlled people, and they are banked into huge stem cell banks, and which can be utilized by scientists to do research, or they can be utilized by companies, pharmaceutical companies, to identify new drugs, or they are used by other companies to test toxins on them. Basically, these iPS cells, like I told you before, they can be used for drug development, disease modeling, regenerative medicine, and to study molecular biology. Large cohorts of patients, cells are collected from these patients, they're converted into iPS cells, and those iPS cells are banked. We have one small bank in Bangalore. It is of all psychological and neural disorders. It was made by a collaboration between NIMHANS, NCBS. What they have done is they have taken cells from a lot of psychological and neurological patients. They've converted them into iPS cells, and now this bank is being very useful in modeling diseases, psychological diseases, psychiatric disorders. I've just got tables of what can be done with these cells. I will read out one example. For example, if you have amyotrophic lateral sclerosis, that is a genetic disorder, there are many genes which are involved in this disorder. And when scientists have modeled this disease from iPS cells of ALS, we have found features like neuronal death, neurite degeneration, reduced soma size, and disturbed mitochondrial morphology, etc., in these cells. So we can find out exactly what is happening at the cellular and molecular level by using these iPS cells. Likewise, for other neurodevelopmental disorders like schizophrenia and Fragile X syndrome, we can also use it for cardiac disorders like hypercardiomyopathy or Leopard syndrome. All of these are genetic disorders. What else can you do with stem cells? So you can model diseases, and there is this never-ending effort to take them to the clinic, and slowly they are taking them to the clinic. What else can you do with these stem cells? Now, in the last 4 years, we can actually convert these stem cells into artificial embryos. So if you treat them with chemicals in the lab and culture them in certain conditions, we can grow them as a ball of cells, which is very, very similar to an embryo. If you want to study what happens in implantation failures or pregnancy failures, you can make these artificial embryos with the genetically disordered stem cells, and you can study what happens. You can take it one step further also. You can allow these artificial embryos to grow in the lab for a certain longer time, and they become the gastruloid stage. That is a one step further in development. These are called gastruloids, which are similar to the gastrular stage of development. And you can study what goes wrong during gastrulation also. Now, mind you, these events are unseen to us because they all happen intrauterine. What happens during these stages of life is completely unknown. Popularly, it is called the black box of development because we don't know what is happening during these stages of life, but we can replicate all of this in vitro with the use of pluripotent stem cells. Apart from this, stem cells are used to make one very important structure called an organoid. An organoid is a miniature of an organ. What is shown in this picture over here is a brain organoid. You can culture it for, if you culture stem cells in certain conditions for 3 to 4 months, it forms a very small structure which is very similar to the brain. If you dissect it and see it, it has all the layers of the brain, it has ventricles of the brain and structures of the brain. This is the recent paper by Gabriel et al. Now you can see these two black structures. These are similar to the eyes. So as time is passing on, scientists are developing closer models which can model closer models to human development. These are, I've just gotten the picture of a brain organoid. You can make lung, liver, heart. All these organoids can be made, and you can study what is happening at the organ level using these organoids. Apart from this, you can use stem cells in tissue culture, which is more applicable to transplantation. I've just got one example over here. What is done, what is this structure? It is a cadaveric heart, human heart. It is decellularized. That means all the cells from the heart were removed by giving some perfusion method. So you remove all the hearts of it and leave only the scaffold of the heart, that is the outer membranes of the heart. This decellularized scaffold, it can be repopulated with stem cells. So after doing that, the scientists repopulated this outer structure with stem cells to see if it can form a complete heart once again. So if you remove the cells from the heart and repopulate it with stem cells, whether it differentiates into the heart. they couldn't achieve the full the difference the generation of the heart to the full extent. But of course, it is one step in going ahead. So you can use it in tissue engineering also. So I've just given a broad in this talk. I just gave a broad outline of what stem cells are, how many types of stem cells are, and what are their major drawbacks, and what they can be used for successfully in today's scientific field. Okay. Now these organoids that I mentioned, they can be made from any. All of these are done. Yeah, there are lung organoids which were very useful in studying COVID infections. There are liver organoids, intestinal organoids, stomach organoids. All of these have been made, and they are all being used to either study infections or dissect some kind of a disease that is happening in the body. This is again what we can do with the organoids. Now, just to say a few highlights of what is happening in the field of stem cell research. People have transplanted neurons into brains, and they see that they do integrate into the circuitry of the brain. People have made iPS cells from endangered species like animals. So to preserve those cells, that can be done. They've made cerebral organoids which are helping in modeling development and microcephaly. Microcephaly related to Zika virus was also modeled using brain organoids. Likewise, there are your Zika virus is written over here. You can model schizophrenia. You can model genetic diseases like thalassemias and anemias with this. Blood cells are more easy to handle. You can differentiate them into blood cells, and because it is a liquid cell type, they can be transfused into human beings, and they can be used for therapy. Now, this is the last type of stem cell that I'm going to talk about. This is called, it is called the mesenchymal stromal cell. It is a mesenchymal cell, as you can see in the picture. These are more popular for transplantation, and there are many institutes in the world which have gone ahead with gone ahead in using this cell type for therapy. It was identified by a scientist called Friedenstein earlier, and he was studying the hematopoietic stem cell. While studying the hematopoietic stem cell, he came across this cell. Now, first, when he isolated this cell, when he isolated the hematopoietic stem cell, there was one type of cell from that group of cells which was attaching to the plate. It was not the hematopoietic stem cell, but another stem cell which was attaching to the plate. And he started studying it further, and he called it the mesenchymal cell. After that, these mesenchymal cells have been isolated from various tissues of the body. You can isolate them from the dental pulp, fetal membranes, bone marrow, umbilical cord, adipose tissue, milk, placenta, like all of these have this mesenchymal stem cell. This mesenchymal stem cell, it can differentiate well into three lineages: adipocytes, chondrocytes, and osteoblasts, but other lineages are a little difficult. That is not the use of this stem cell. This is the use of this stem cell. Now, this cell, when injected into patient, it has an immunomodulatory effect. So it is known to somehow manage the disease much better. It plays a significant role in tissue repair and regeneration. It has immunomodulating responses, and it enables immune suppression and tolerance induction. And it is being successfully used across the world for many indications. In India, in Bangalore, we have a company, the Manipal group has a company called Stempeutics. Stempeutics manufactures these cells, meaning they take it from the donors, they upscale them, and they have used it very well to treat osteoarthritis. So it is giving very good results with that. It is very good for diabetic foot ulcers. So a wound that was not getting cured by other methods has shown very good recovery with these cells, and they are using it for critical limb ischemia also. So as of now, as of today, mesenchymal stem cells are already into the market for transplantation. Pluripotent stem cells and other adult stem cells which are isolated from other organs need more. The protocols have to be defined better for them to be used as stem cells on patients. That's about my talk. We at MIRM, we do some basic research on stem cells. I have gotten what I do in my group. I work basically with induced pluripotent stem cells. We are refining the methods by which we can differentiate cells into different types. We are trying to study what are the shapes of cells. You all might not be interested in it, but what each cell type in the body has a different shape, and each shape has a different function in that tissue. We are trying to understand what happened, what brings about these shape shifts, or what is responsible for one cell having one type of cell. We are modeling syndromic bicuspid aortic valve disease by using iPS cells derived from patients who have the bicuspid aortic valve disease. We are modeling neural tube defects which are caused due to folic acid deficiency by generating organoids, and we culture them in absence of folic acid, and we try to understand what exactly causes the neural tube defects. And we are also making artificial embryos to understand pregnancy failure, like what exactly aneuploidy syndromes or aneuploid cells, they are more prevalent in pregnancy. I mean, they're responsible for pregnancy failures. So we do that work also in my lab. And of course, we study the minor aspects of cells like cell polarization, what happens with these embryonic stem cells and stuff like that. I come from a place called Manipal Institute of Regenerative Medicine. Just as a background, I'll tell you all, we have these following, we have only one course or one subject. We teach stem cell biology, and we teach stem cell technology and regenerative biology. We have 4 different courses: MSc, Advanced Diploma, MPhil, and we have PhD courses. And we are all working towards providing a highly skilled workforce in the stem cell field. These are all my colleagues in MIRM. This is Dr. Sudha Warrior. She's the Dean of MIRM. Her major interest is cancer stem cells, and she works on signaling pathways which make cells cancerous and how to handle the cancer stem cell. This is Dr. Jyoti. She works on mesenchymal stem cells and the immunomodulatory properties of mesenchymal stem cells. Dr. Anujit works on iPS cells derived from Alzheimer's, and he's Alzheimer's and Parkinson's, and he's finding out what is wrong with those cells. Dr. Anand works on epileptic models of stem cells. Manasa is a tissue engineer, and she's engineering the uterine walls and other tissue engineer scaffolds. Dr. Sangeeta works on Parkinson's and Alzheimer's also. Dr. Uttara works on mesenchymal stem cells and how fungal infections affect different parts of the body. Meenal works on diabetes. So she's isolating islets, and she's finding newer methods to cure diabetes. So these are some areas that we research in MIR. I thank Dr. Vinod Bhandari, Dr. Vavare, Jayashree, Dr. Jayashree, Deepa, Pankaj. I couldn't meet Pankaj for inviting me to science to interact with them. Yeah.
Thank you, ma'am, it was very informative talk. We can now take up a couple of questions. Any queries from the audience? Any queries? Yes.
No question.
It's
Thank you very much for the informative talk. So I just wanted to know, you have mentioned the matured cell can be converted into stem cells by introducing 4 genes.
Yes.
So my perception is the matured cells are coming from the stem cells only.
No.
They will be having the genes within them.
No.
When the cell goes on dividing, the genes will be carried forward from one cell to the other.
No, the mature cell does not have those 4 genes because it is already mature. These are pluripotency factors. So they are not present in mature cells like fibroblasts or skin or blood. We have to introduce them from outside.
Okay.
Once we introduce them, they reprogram the cell.
So you mean the pluripotent stem cells, as they go on dividing, they will lose those genes?
They will lose the genes. They will lose their pluripotency and they become more specialized. Pluripotent stem cells are present only in the embryos, in the blastocyst. As the blastocysts develop, it loses its pluripotency. It becomes more specialized and more specialized and more specialized. So eventually, adult cells in our body are not pluripotent anymore. They are all specialized now, and they don't have those factors. So if you want to convert that back into a pluripotent like state, we have to forcefully introduce those genes outside, from outside.
Thank you.
Any other queries from the students? Students, any queries? Yes. Any queries?
Are just storing their child stem cells or ऐसे कोई प्रॉब्लम होती है तो जैसे लिवर डैमेज है तो उसको दोबारा रीजनरेट कर सकते हैं इसका फ्यूचर रीजनरेटिव डिजीज का कैसे होगा?
Procedure to store the cord blood stem cells after delivery. Cord blood stem cells can be used for other interventions, like Dr. Jyoti was mentioning, for other oncology related. We do get some results there. Saying that if you store a child's cells and those cells in the future काम आएगा, depends like what exactly the child is going to go on and what is the disease that he is going to go through. Definitely, it cannot be an immediate cure for accidents or injuries, traumatic or acute injuries. Depends on what like you can just inject, मतलब अगर कोई accident हो गया और कुछ brain damage, so you cannot take those contrast and it's not going to help as well. इसके लिए थोड़ा
Thank you, ma'am. Any other queries? Thank you, ma'am. With this, we conclude this session. I thank you all for the active participation, and we would reconvene at 2:15 p.m. at Laser Auditorium. Thank you. Kindly submit the feedback form at the reception.
First.